Thank you so much. It's such a pleasure to be here today and to share with you some really exciting new research that we're doing in my lab right now. I want to just first off acknowledge, though, that I'm going to show you a lot of data that is the work of a lot of people, and I feel like one of the greatest privileges that I have in the kind of work that I do is that we get to do team science that incorporates the talents of so many people. We're often trying to tackle really hard problems that have no clear solution, and so we have to be very creative about bringing in people with very different types of expertise to try to reconstruct these deep histories in human evolution. But today I'm going to focus the work on just a few of the people in my lab, some postdocs and PhD students who I'm going to really feature today, whose work has contributed to our study of the evolution of the human microbiome. And I want to make an argument that I think why it's so important to study the human microbiome is that I don't think we can fully understand what makes us human without also considering our microbial self, because they perform so many of the key functions of our biology that they really are a part of us. When we think about where we're going to kind of study the microbiome, of course, in a fully living human, we have many different habitats across our body, different microbiota. But when it comes to the archaeological record, our best source to understand the long coevolution of these microbes is teeth. Teeth are really amazing. This is an example of a tooth from a woman who lived about 1,000 years ago in Germany. I'm just showing it here in cross-section. And we have this accretion of dental bacteria that begin as plaque and mineralize during life to form tooth tartar or calculus that is a really rich source of the oral microbiome for us. And we've been able to reconstruct bacterial genomes going back as far as Neanderthals. But importantly, it also encapsulates other information about the types of foods that we have eaten through time, for example, through starch granules or even food proteins. We've been able to use proteomic analysis of calculus to study the spread of milking technologies, for example. Teeth are also really important for studying ancient pathogens because any bloodborne infection that a person has at the time of death, some traces of those pathogens will also be present in the pulp chamber of the teeth, which is vascularized and has a lot of tissue. And so the teeth act as this preservation environment where we can often recover some of those infectious pathogens from the past. And of course, teeth are also fantastic for reconstructing the human genome, and that's very useful if we also want to study the spread of microbes, because the people are the ones who move them around. Now, teeth are not the only source of ancient microbiota that we can study in the past. We do, under certain extraordinary conditions, also find paleofeces, and we can use this to reconstruct aspects of diet and gastrointestinal health and gut microbiome evolution. And in some cases, we also find microbial remains in large vessels like this copper cauldron you see here, from which we're reconstructing some of the early beer ferments from the Himalayan mountains, or from large pottery vessels like you see here at right, which is a big Roman wine fermentation vessel where we're trying to understand early wine ferments. The biggest problem that we have when we work with ancient microbial DNA, of course, is that we're not working with pristine, intact genomes, but rather the shattered, fractured remains that are left over time. And so for an ancient microbial genome, which is on average about 3 million bases long, which is much smaller than, say, the human genome, it presents a sort of puzzle for us. So you can think about an average ancient bacterial genome being like a 60,000 piece jigsaw puzzle. So I often refer to ancient DNA as the worst jigsaw puzzle in the world. But it's a little bit more complicated when we're studying microbiota. So I'm going to use a metaphor. I'm going to continue my puzzle metaphor here. And let's just start with an example of, for example, some paleofeces here. If we were to analyze this, what we might find is not, of course, just one puzzle, but many. Within it, we're going to have a human genome. Maybe we have a pathogen genome, something like Salmonella enterica. And then we're going to find lots of other genomes that are related to the microbiome, but also the necrobiome, those environmental microbes that invade this after death and also leave behind their DNA. Now, it would be great if this was all organized like it is in this toy shop for us, but when we receive it, it's kind of more in this state, so all the boxes have been opened, all the puzzles have been dumped on the floor, and to make matters worse, most of the boxes have been thrown away, and so we only know what some of them even are, and many we've never seen, and have no idea how many microbes we're actually trying to reconstruct or what many of them might have been. Just to use another metaphor, one that's used a lot in microbiome sciences is that of a coral reef. So microbiota are often compared to coral reefs because they're these diverse ecologies of many species that interact with each other and are often codependent on one another. And so if we think of a coral reef as this past microbial diversity that we want to recover, you can think of parrotfish as time. Parrotfish go through and they eat that coral reef and they leave behind white sand. And so the problem is not that this ancient DNA is no longer with us. In fact, we're completely surrounded by ancient microbial DNA. The question is, how do we put it back together to reconstruct those ecologies and make sense of them? So we began this work trying to understand the evolution of ancient human microbiota by focusing on teeth and focusing on the oral microbiome way back in 2011. This is the same individual I showed you before. We're going to take a closer look at her teeth, and we can zoom in more on this dental biofilm, this calcified dental biofilm called dental calculus. What's really amazing is you can see that it's structured, it's laminated, and that's because it forms incrementally. So you have a layer of dental plaque, which is a living, growing biofilm of bacteria. It will spontaneously calcify, and then a new layer of plaque will form, and this happens over and over again throughout the lifetime of an individual. It's not quite annual, it's semi-annual, but it builds up over time, much like rings of an onion or layers of an onion or rings of a tree. And it preserves in exquisite detail the oral microbes that were once there. So we can zoom in a little bit here and see, for example, under higher magnification, that these bacteria have been just frozen in place through these what are rather rapid mineralization events. We can demineralize them, we can gram stain them, we can stain them with dyes that make the DNA visible and see actually they're incredibly rich in DNA. We can also visualize the proteins that are inside. This is using immunohistochemistry, showing they still have functional epitopes. So what we have here is an entire frozen system in time. Dental calculus turns out to be the only part of your body that naturally fossilizes while you're still alive. And so it preserves these molecules in a manner that doesn't undergo the same decomposition processes of the rest of your body and allows us to study them into deep time. Our first paper on this was in 2014, and we were able to use genetic technologies to reconstruct many of the major members of the oral microbiome that you see listed here, and we were even able to reconstruct the first genome from an ancient microbiota, and that was in 2014, which was just 3 years after Johannes and his team led the first successful reconstruction of an ancient bacterial genome, which was Yersinia pestis. And so we turned to this to try to understand from this really rich source of ancient microbiota, could we then reconstruct aspects of the evolution of our microbiome? As we dug into this problem, and the first thing we wanted to really do was place this with an evolutionary framework, because up until this point, primates themselves hadn't also really been studied very much. We wanted to understand how similar or different was the human oral microbiome. So we began this, we looked at the different African great apes and also howler monkeys as an outgroup, and we collected a lot of dental calculus from ancient humans throughout the Holocene, the Pleistocene, and Neanderthals. We had many findings in trying to compare this and look at how the human oral microbiome has changed through time. I only have time to highlight a couple aspects here, but ones that I think are really exciting. One thing that jumped out immediately is that humans are really different in how our oral microbes process carbohydrates. And this caught my attention because there's a lot of interest in trying to understand early human diets. We can dig into that and we can kind of find out what's actually driving some of these differences, and it turns out that it's actually mostly one group of bacteria within humans, and this is a group called Streptococcus. Now, it's long been known that Streptococcus are the major bacteria in the human oral cavity, and it was largely assumed that was just true in general, perhaps for mammals. But what we now know is that actually Streptococci are not very common in the oral microbiome. This is a more human phenomenon. In fact, they're actually not that common in other primates. However, within Streptococcus, there are many different types, and here's some of the groups that are common. What we can do is look to see which ones are present in different primates and in humans. So these are all scaled up to 100%, so not all of them actually have very many streptococci, but you're looking at the proportions that they have of these different groups. We can look at howler monkeys here, which have a relatively even distribution of some of these different groups, and gorillas are a little bit more variable. Then we have chimpanzees, which are really odd because they're completely dominated by the anginosus group, and then humans show a very different phenotype. So we're really strongly dominated in most individuals with this group called the sanguinous group, and to a lesser extent, the mitis group. Now, this group is really interesting because they have a property that no other bacteria in the mouth have. They have a series of genes called amylase binding protein genes, which they can use to actually steal the amylase out of our saliva, which is an enzyme used to digest starches. So they've figured out a way to hijack our enzymes to help themselves, which is really interesting. Amylase binding protein A and B are two genes that encode this, and they actually work together synergistically to give these streptococci a huge advantage in a starch-eating population. First of all, a metabolic advantage, because they're the only ones that can actually then take advantage of the dietary starches that may be coming in. The other microbes actually lack the ability to break down those large crystalline structures. But I think probably even more important, it gives them a colonization advantage. It turns out most bacteria can't actually stick to the surface of our teeth. They slide right off, and there's only a couple groups that can. And that's because they've evolved receptors that bind onto something called the acquired enamel pellicle. This is a thin layer of proteins from saliva that coat our teeth, and it turns out salivary amylase is one of the major components of the acquired enamel pellicle. And by having this receptor, streptococci are able to bind on better to the teeth and therefore initiate biofilm formation. So we saw that these groups are different across primates, and we wanted to know, though, are the genes present or not? And so we compared this across non-human primates and within Homo, and what we found was that the amylase binding protein A and B are extraordinarily low and largely absent. These are probably just spurious mapping that we have here in the primates, but they're high in Homo. What was really interesting is we expected this, of course, for anatomically modern humans, but what we didn't expect is they're also present in Neanderthals. This, to me, immediately raised some questions about early human diet and made me recall some early work done by Leslie Aiello, who posed the question of the expensive tissue hypothesis. She noted that early in human evolution, one of the things that differs is that from us and chimpanzees is we shrink our gastrointestinal tract at the same time we're growing a large brain. And she thought that the only way this could be explained is if part of what's contributing to the formation of Homo and to the formation of modern humans is a dietary shift to something that's much more energetically dense. But what could that be? A lot of hypotheses were proposed, meat consumption and cooperative hunting, for example, but Richard Wrangham had proposed that perhaps it's the invention of fire and cooking, and specifically cooked starches, which are widely consumed and widely available throughout the kinds of environments where early humans were evolving, might be the answer. But it's very hard to find plants in the archaeological record. What we might have here with amylase binding proteins, that genes that are evolving within the human lineage, is a clue to some of these early human diets that perhaps starch was very important. And we may be able to get at it by studying the evolutionary history of the acquisition and diversification of some of these genes. Now, one of the other things that we found, we were able to also establish a core oral microbiome, so what are the core members that everybody has, and we were able to identify a number of different genera that have been conserved within the human lineage for more than 40,000 years. But one of the things that was very surprising to us is actually at least half of those are very poorly studied, nearly unstudied within the dental literature, in part because they don't cause disease, so they hadn't been a high focus of research. We know very little about them, and this gave us a lot of problems. How do we study something that has almost no reference genomes? In fact, for one of these, there's an entire class of bacteria for which only one reference genome was available. And so we have such limited diversity in our comparative data, we were having a very hard time exploring this in the past. So how do we then reconstruct an ecology if we know that our modern references are lacking most of the members? In 2020, we started to tackle this problem using a new technique that became available, which is called de novo metagenomic assembly. And the idea behind this is if you sequence deeply enough, you can actually just use the DNA itself to overlap the pieces to start to reconstruct de novo genomes with no comparative reference. We were able to successfully apply this to some paleofeces that we analyzed, and were able to reconstruct both known and novel species that did not exist in current databases. Now, this was very exciting, but of course the next question you want to ask is it real? Are these chimeras? How much can we trust this? And so we thought we would turn to a problem that could answer this. We said, well, let's start with something hard. Let's start with Pleistocene era microbiota, so we'll start with Neanderthals and Pleistocene era humans. We're going to reconstruct these genomes. We're going to look for a particular class of genes called biosynthetic gene clusters. These are groups of genes that together encode enzymes that work as a machine to produce natural products. This is important because if there's any error in the code that you've reconstructed, those enzymes, those machines won't work and they won't make the final product. And so we were able to reconstruct a number of genomes, identify a whole series of biosynthetic gene clusters, and identify one in particular that was really, really widespread across many individuals in the Pleistocene. We were then able to show that we had a high conservation of gene synteny, for example, in these biosynthetic gene clusters. We were able to then synthesize them, put them into living bacteria, and express them to see if they would make the predicted product. They did, they made the correct product, and this was a confirmation that our assembly methods were accurate. We then went back throughout the entire record. We assembled as much data as we could from all the existing published literature, also building databases to try to gather all of the ancient microbiota that had been published. We were able to find about 14,000 oral genomes that had been published in various sources, and then we were able to take metagenomic data that had been sequenced from various saliva studies, plaque studies, and ancient dental calculus studies all over the world, and then add to it new studies that we did in novel populations that hadn't been explored before. From this, we were able to assemble an additional over 10,000 new genomes, of which more than 3,000 come from ancient dental calculus. And together, this is a new genome catalog of nearly 25,000 oral microbial genomes. And you can see also we span about 102,000 years of time. We actually have quite a few representatives from early anatomically modern humans and also from Neanderthals. So when we take this all together, now we can cluster them into species clusters and ask how diverse is the human oral microbiome? So this was the state before we began. There were about 500 known, well-described oral species in the human mouth, and this is just representing them on a tree of life. And adding in our new genomes and clustering them to species, we can dramatically expand this. We've now expanded this to approximately 3,400 oral species. Much of this new diversity is actually added by ancient dental calculus, which kind of extends the diversity that is known within these dental biofilms. And we can also perform another type of analysis. We can look at differential abundance analysis. So we can say, are there things that are dramatically changing, that are really different between, for example, people living in industrialized societies today and people living in the past? And this is just a volcano plot looking at this. And we can apply a type of analysis called vanished blossom analysis to look at taxa that are vanishing, that are massively declining. This stands for volatile and or associated negatively with industrialized societies of humans. And we can look at things that are blooming or bloom or selected in societies of urbanization and modernization. And these are two different categories that have been explored a lot in the gut microbiome. And so we applied this to the oral microbiome. We asked, are there things that are either massively declining or increasing between ancient humans and today? We find that there are actually quite a few that are changing to a large degree. What's really interesting is the things that are blooming are really well-known, well-characterized taxa, some of which are actually associated with disease. And many of the ones that are vanishing are poorly known, have no names, don't even have genera names for them. We can visualize this just differently so you can kind of see the large differences here. On the left, you see everything that's a dark blue is present in, say, ancient calculus, and is absent in light blue. So you can see on the far right, we have industrial plaque. That's like our plaque. We have a really different kind of spectrum of species that are present. In fact, we've lost many of these species that are widely spread across time in ancient individuals. I'm going to just dive into one of those tiny rows that you see there. This is one we've been really interested in. This is the Neanderthal family. It has quite a few different members that are known to live in the human oral cavity, although all of them were poorly studied. We don't really know what they do or what their function is. And there's one that right here, we've given it a candidatus name. It didn't have a name, so we gave it one, Cryptocalculus ophthonis. It turns out that it is the most abundant and most prevalent oral species up to 200 years ago. And today it is hardly found at all. We see it as of 200 years ago and before. We have global distribution. It's found over wide ranges of time. It's also found in Neanderthals. And what's really amazing is that we can not only say it's there, but we can say a lot about what its functions might have been. So for example, we've been able to reconstruct a complete vancomycin operon, I think it's vancomycin resistance operon within this species, and trace its evolution from the Pleistocene onwards. It actually retains this gene synteny amazingly well over that time period. We've also investigated its CRISPR-Cas system. It actually has a novel one that hasn't been described before, which we're really excited to dig further into. We can look at its changing proportions through time and show that it's actually quite low in the deep past, and then it rises steadily through time. It actually reaches its peak in the 19th century and then completely crashes in populations today. And we don't know why. And it's not just this species. You can take any one of those other species and make similar plots showing dramatic changes through time that have occurred. Now, I don't want to end without saying some more about Neanderthals, because we had so many great talks this morning. We have genomes now reconstructed for 47 different Neanderthal oral species, most of which are actually shared with humans, although there are 12 that are so far unique to the Neanderthals. When they do appear when humans and Neanderthals do share a species, the Neanderthals often do form their own clade, like what you see here in red. But in a few cases, we actually see that humans and Neanderthals form a clade together. When that does occur, it's Upper Paleolithic humans. We think this may be evidence of past contact. We know that the oral microbiome is transmitted through intimate contact and cohabitation, and so may reflect some of these really early interactions. So I'm very excited that I think that ancient DNA is now allowing us to reveal this evolutionary history of more than 3,000 of our microbial species that are today known only from microscopic fossils. This is an archaeology of the invisible that is revealing our microbial self, and I'm hopeful will give us a lot of insights in some of the important invisible behaviors of the past that are very hard to study from the material record, but may be encoded in these microbial genomes. Thank you very much for your attention.